The overall goal of this procedure is to increase specificity of antibodies against closely related proteins by removing cross-reactive antibodies. This is accomplished by first identifying protein domains of greatest divergence between homologous proteins. The second step is to express and purify these protein domains. Next, these protein domains are coupled via the N or C terminal to a chromatography matrix. Cephalose is used in this demonstration. The final step consists of incubating immune serum with resins coupled to each cross-reactive domain. Ultimately, ELISA and Western blotting are performed to assess the efficacy of the procedure.
The main advantage of this technique over existing methods, like the use of monoclonal antibodies or affinity purification, is that we can maintain affinity against the desired target while eliminating known cross-reactive epitopes. Generally, individuals new to this method will struggle because the protein they're trying to study is unstable. Demonstrating this procedure will be Dr. Woori Kim, a postdoctoral fellow from our laboratory.
This study requires two columns, one for neuron-derived orphan receptor 1, or Nurr-1, and the other for nerve growth factor IB, or Nurr-77. Since the same protocol applies to both proteins, only the preparation of one column will be demonstrated. Thaw the protein to be coupled to the resin on ice, and then determine the protein concentration using its